In this video, we will discuss about how we can use this microfluidic to measure the wound closure assay. So these are the steps that I mentioned here, and we will follow this step. But however, we will include one more additional step so that we will be able to know what kind of cells that favors this wound closure assay. So let us start with the first step. So in this case, first is we will wash the microfluidics with the help of this PBS buffer, and after that we will keep this microfluidic in the standard cell culture condition, that is 37 degree and 5% CO2, and thereafter after 1 or 2 hours we will change this buffer from PBS into the DMEM plus this 10% CS, or it's not necessary to be 10% CS. We can use DMEM plus FBS plus antacid, which is right now the market standards for the regular cell culture medium. So when the this microfluidics will get saturated with this cell culture medium, we can seed the cells with the help of the injection. So in this case, in the paper it is mentioned that we can use 3 into 10 raised to power 6 cells for this one microfluidic device, and we will continue to pass the DMEM medium till this monolayer of the cells has been observed, till this monolayer of the cells has been acquired. And thereafter what we will do is, since there are 3 different channels, from one channels it can flow into the middle line, and from the other channel it divides into the 2 side lines that can be seen here. So in this till this stage, when the cells are growing, DMEM is only flowing through this medium, only flowing through this central medium, and helping the cells to get their monolayer. And thereafter what we will do is we will switch the case, that is DMEM will now starts to flow into the corner, and the trypsin will starts to flow into the middle line, so that the cells in the middle lines will get removed from the cell surface, and wound closure assay can be started, we can say that. However, in this case, when the cells are get stripped off from this middle line, what we can do is we can barcode our these cells that are on the corner, so that now we will have the cells which are uniquely barcoded. And thereafter what we will do is we will stop this trypsin, and we will stop this trypsin from the medium cell on the medium channel, and again restart the DMEM flow from this medium channel till we get the we can say again monolayer, till we get the monolayer again. So in this case, the cells that helps in this wound closure will then can be analyzed with the help of barcode that are present on the cells, which are labeled during this treatment stage. So in this case, we will not only have the wound closure assay effect, but also we will be able to know which are the cells that helps in this wound closure. And this kind of experiment is usually useful when we have some kind of knockout library, and we need to check which knockout cells accelerated this wound closure assay or deaccelerated the wound closure assay. And the actual image is shown on the left hand side. Then this at t is equal to 0 hour, the image shows is resembles with this treatment stage when the trypsins are removed from the middle region. middle line, and after this 24 hours we can see that the cells starts to cover up this middle region. Cell starts to cover up this middle region, and the distance between two cells, distance between or the width of this middle channel decreases in the T24 hour image. So this indicates that wound closure assay is going on even after this 24 hour, but it is not yet completed. So in this paper they have not used any barcode because they have not used any kind of knockout library, but this barcode idea can be used if we are using some kind of knockout library and need to check which cells favors this wound closure assay and which cells does not favor this wound closure assay. So I hope you have understood how we can use this microfluidics to measure wound closure assay, and even we have gone one step further to check out of a knockout library which cells promotes this wound closure assay and which cells do not promote this wound closure assay. If you need to understand more about this microfluidics wound closure assay, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.